So, good morning again. For those of you few brave souls which are still here. That's right. So, we discussed a few things so far. We have considered one very simple network that can do predictive coding without any supervision. And that's how the brain learns, right? Unlike supervised models where somebody's out there telling the neurons or weights what to do, that doesn't happen in the brain. There is nobody telling them what to do. And still, we are pretty good. So far, we are beating the machines. So we want to understand what is that secret sauce. How is it that the brain is doing that? And I suggested in order to do that, we have to understand a few key principles of how that circuit works. The circuit is very complicated. There are lots of things going on. We can't keep track of everything, so we are going to try to become something like Kepler and figure out some fundamental rules of what's going on in the brain and how does it do that. So this was one attempt to say, look, I'm going to ignore a ton of things. I'm going to imagine there is only one output neuron getting a bunch of inputs. This much is certainly true. More may be going on. This may be connected to other neurons. We're going to imagine the rat's running with a uniform speed. Clearly, the rat doesn't do that. We're going to worry about that later. And all we did is we simply put in that biological learning rule, and this thing generated successor representation very, very clearly in that simple one-dimensional task. And then we said, if that is true, it should actually happen biologically, because lots of things happen in biology. And we found, lo and behold, it does happen, that in realistic neurons, as real as it gets, within a few trials, the neurons' receptive fields become wider, they become asymmetric, and they develop successor representation. So far, so good. So let me summarize this, that what did we learn so far? What are the few key points that I can try to convey so far? First, I conveyed that unlike computers, where nodes are just threshold linear, neurons actually generate spikes. Key feature. Neurons are of two types, excitatory and inhibitory. So far, we only worried about excitatory neurons. And so far, we even ignored the fact that neurons generate spikes because can't do everything. We assumed that neurons are smooth, even though we know perfectly well they are not smooth. We know that brain has unsupervised learning rule, and that unsupervised learning rule can generate predictive coding in smooth neurons. That's what I showed so far, and that seems to match with the data amazingly well. So, pretty good. Any doubts about this? This much that I covered in 2 lectures? Why did it take me so long? So now let's move forward with this. I'm not going to go through various advantages and so on. I can show this slide. There are many predictions that this model makes, which have been verified because you don't want to just do one thing and other things. So there are many, many tests that I'm not going to go through. But now let me show you why. Let's start going closer to biology. So when I want to get closer to biology, I want to bring in 2 facts. Yes, please.
Could you go back to the previous, the summary slide again?
Yeah.
I was just going to ask, what...
This one?
No, sorry, back one again. Were you just summarizing what we went over, I guess?
Yeah.
What did we learn? How do we, what price do we pay, if any, for making the idealization that neurons are smooth, that they're not?
We are going to discuss exactly that.
Okay.
That's exactly what we're going to do. All right? Any other questions? So we want to know how much of that, the surprising thing is that even though we made lots of assumptions, we ignored spikes, we ignored inhibition, it still works somehow. And I gave you a big slide full of things why it works, not just 2 or 3 observations. So the cool thing is that the model has only 2 assumptions which are solidly verified. Each neuron is activated by 10 or 20 neurons at the same time, otherwise it doesn't activate, and the learning rule has this temporal asymmetry. It's well established, you just put it in, and it does it, and it matches this 20 or so observations in different things. Incredible amount of success from an incredibly obvious model, you could say in hindsight. So why? And what can we learn? Yeah? Smooth meaning there are no spikes, right? So I just computed a smooth curve. When I showed predictive coding, each neuron was just a smooth function. There were no spikes there. But that's where we started, right? Everybody cool with that? So that's what we did. And I'm not going to go through all this. No, let me not go there. Otherwise, you're not supposed to know any of this stuff. Yeah. Let me unhide this because it looks like all this stuff should be open. All right. So these are the set of things that I'm not going to tell you, but they are there. If you want, you can look up these references. There are lots of verification of this simple idea. Yes, please.
So in some of these slides, you used the word, and I apologize, I wasn't here for your previous talks, but anticipatory and predictive.
Yeah.
And from a psychological perspective, I think of those as kind of different things.
Yeah. This is, yeah, I'm using it. It's extremely simple right now. We want to go towards what is anticipation, what's causality, what is predictive coding. Right now, I just have a little arrow between just a handful of neurons.
Okay.
But I'm going to try to show you, depending on how far you go, that it can do all these things. In fact, it can do other things that you're doing all the time. You're doing it right now, actually, which you don't think you're doing. So maybe I'll do a demo here, live demo, all right? So I'm going to stand in front of you. You are the participant, and I'm going to simply move my finger in front of you, and I'm going to ask you to catch it as soon as I say go. So go.
Oh, wait, I'm sorry. You want me to...
You're going to just physically grab my hand as soon as I say go. So all right, 1, 2, 3, go. You did it. Super, right? Now let me show you that your brain is doing this right now. So what is it that happened in this amazingly simple task? Your eyes saw some stimulus, and I said go, and you went towards it. But stimulus is moving at 1 meter per second. You should have gone 25 centimeters behind it. You didn't do that. So even in the simplest task, such as just grabbing something, your brain actually created a prediction. So the hand is moving at this speed, it's going to be there, and you went there. You think you went there, you didn't. You actually went there. So we are doing this constantly. So we have, in psychological terms, we have come up with different names. Something is this, something is that, but this is the kind of memory that's going on all the time. You're not even aware of it. That's why you go and stand in front of a cliff, and you start feeling this weird sensation at the back of your leg. It's your brain saying, dude, bad idea. And you don't know that, you don't hear the sound, it's pulling you back. I ask you another question, you lift your hand. You can do that experiment yourself. Lift your hand, what's the first muscle that moved? What's the first muscle that moved when you lifted your hand? I have already primed you, but you won't feel it. If you lifted your hand, it's not the elbow, it's not the shoulder, it's the muscle at the back to keep your center of gravity. You're highly unstable. You're going to fall on your nose. If the first muscle at the back didn't move and pull you back, so the center of gravity remained, you're going to topple over. So you're doing this constantly. Even when I told you you're doing it, you don't feel it, because it's very, very fast. It's all been hidden. And that's why babies, when they walk, they're like doing something like that, because they haven't learned those things yet. It hasn't become automatic. So this is going on all the time. And this, I suggest, is at the core of all sorts of machines, and these are various evidences from lots of places. But now let's go closer to biology to say, okay, can we go slightly beyond that and get to somewhat realistic neurons? And there is this weird thing called attention, right? And we have papers, attention is all you need, but what is that thing called attention? So that if you don't pay attention, you don't learn. What's all about that? How does that work? So let's start with this. So we summarize everything that we stated so far, that learning by this Hebbian plasticity, also called spike timing dependent plasticity, requires precise spike timing between the presynaptic neuron and postsynaptic neurons. The input and the so-called output neuron should be fired in a precise temporal order. That's what is needed. If they do something by 100 millisecond jitter, everything is gone. 10 millisecond high precision, that's what is needed. So we studied this so far. That's what we studied. So if you have time, and if you have smoothly active neurons, then you will generate the right kind of prediction, and predictive coding will work, and all that stuff. So that's what I showed so far. This was a huge approximation, and on the y-axis is the probability of spiking, some smooth function. But that's not what neurons do. And now, for the first time, we have to worry about time. So this window has appeared called 100 millisecond, more or less. It's a cartoon scale bar. Now, what a neuron will do actually is this. That neuron is going to generate spikes, technical term, firing rate modulated inhomogeneous Poisson process. So at every moment, neuron tosses a coin, whether to generate spike or not. The probability of yes is a rate function, and this is your rate function. The rate function is constant. It will keep generating spike in one exponential distribution of Poisson process. So some more spikes will appear here, and less spikes will appear here. That's what it will do. And in a different trial, more or less the same distribution will be true, but this spike may be here, that may be there. And we know, here is my time to jostle your memory, how variable are neuron spike timing. At the end of my first talk, I mentioned how much is the variability of neural activity. Yes, please. Exactly. Thank you. The jitter in neural spiking activity is equal to its mean, massive, standard deviation equal to mean, and that's why people think neurons encode information by a Poisson process, because that's what happens for Poisson process as well. Standard deviation is roughly equal to the mean. So this is more complicated, but that's a pretty good approximation. So now that's what one neuron does, and that's what the other neuron will do. All right? Now, we have to pay attention to these 100 milliseconds. What that says is that all that action has to happen such that whenever there is the first neuron, red spike, the blue spike should occur immediately after that within 10 milliseconds, more or less, given that's 100 milliseconds. So here there is red, then there is blue, here the red, blue, so something will happen here. This also works, but who is there? Who is there to control this? Because most natural thing that will happen is this. You still have more red spikes here, you still have more blue spikes here, but the most likely scenario is actually this. Sometimes the blue spike is too far from red spikes, given that neurons fire only one spike per second. Sometimes the blue spike is sandwiched between the red spikes. That means this leads to the right thing and this leads to the wrong thing, the two cancel. And sometimes it leads to the wrong thing. The blue appears before the red in that short window. Right? So if you just generate two random time series modulated by this, there is nothing that's going to ensure that the two spikes occur in the same sequence as the behavior. What does that mean in technical terms? That there is certain behavioral time scale. The behavioral time scale is our behavior slow time scale. I'm walking from here to there. It takes me, God knows, 1 second to go from here to there. But neurons are much faster. They're generating spikes with millisecond precision. And the learning rule that we looked at is precise also within 10 milliseconds. But for some reason, neurons, when they generate spikes, they don't respect this thing. That means that everything that I told you crashes, doesn't work. If I had done that calculation just now, with these spiking neurons, there'll be no predictive coding. You'll not be able to catch my hand, and if I lifted my hand, I'll fall on my nose. If we put it in different ways, simple causality, this was tiger, and then you got whacked. This says tiger came before me as well as after I got whacked. Here, the tiger was too far, and here, first I got whacked, and then there was a tiger. anti-causality. Just hopeless. You're not going to learn. So that says that if neurons encode information with only mean firing rate, called rate modulated Poisson process, the precise spike timing that is required for this kind of biological learning will occur rarely, resulting in poor or no learning. We have a serious problem. All right? That's exactly right. And we agreed that every neuron needs input from at least 10 other neurons. So if you're just one neuron activating, yes, no problem. But we know that we need at least 10 or more. And we know biologically every neuron gets 10,000 inputs, not just 10. So that probability is exceedingly low, right? No, you're not convinced. Happy to discuss.
Well, I
don't
mean,
know.
you know, it could be, for instance, that yes, the rate is affected by, you know, you need at least 10 inputs to up the rate, but the precise timing of the spike would be instigated by the fact that there's an incoming spike, just one.
But that's not enough. One spike, when one neuron fires a spike, when that spike...
Imagine there were 10 spikes that just came in, and then there's just one extra.
That's right.
That might just tip it over.
That's right. So then, let's imagine a hypothetical. So now you have to look at a group of at least 10 neurons. Who is going to be controlling all of them? Which one is going to be precise? So you can even imagine, let's be even more generous to say, okay, those other 10 neurons are conspiring. It's just the 11th neuron that does the right thing. That's still pretty bad. Because all the wrong things will still happen. Only one will have control. So it's a serious problem. We have done simulations and so on. It doesn't work, actually. It falls massively bad. So how does the biology solve this? And can this be resurrected? And maybe it solves something else. Now, what that something else is that you want to resurrect is the fact that you don't want to learn everything. Because I'm walking around in the world and I don't want to learn every single thing, otherwise I'm overloaded. That's why we pay something called attention. So right now you have some idea of what I'm saying, but you don't know what's going on. I don't know, maybe there's some ant crawling around or some noise in the room. You don't know those things. You have to ignore all those signals from the rest of the body. That's roughly called attention, but what the hell is attention and how does the brain do that? So we looked at this thing. It's a complicated slide, let me not go through it. Bottom line is that so far we have only looking at excitatory neuron, but there's a massive amount of inhibition. The inhibitory neurons, even though they are small in number, they speak very loudly and they shut down this thing. So that's what happens. So we're going to say, all right, here's a CA1 neuron. And let's start with the fact that it's already getting asymmetric input. We're going to do the recursive thing later. So just for the pictorial thing, let's imagine for some reason this neuron is getting already asymmetric input. Stay with me. Something is already formed that learning. And we are still going to solve this problem. And then we are going to come back to see how it can be solved recursively without going through equations. So that neuron we know, while it's not connected to other excitatory neurons, biological fact, every single excitatory neuron is connected to an inhibitory neuron. That much is true. And every single excitatory neuron gets inhibition. We ignored this inhibition so far because it's too complicated. So let's now put that back in. So that's there. Not only that, what people have found is that as soon as you start paying attention, depending on what kind of attention and so on, the brain starts to generate some different kinds of rhythmic activity. The rhythmic activity appears on these inhibitory neurons. And you can imagine a very simple mechanism. If this is an excitatory neuron and an inhibitory neuron, if this excitatory neuron gets activated, it will activate the inhibitory neuron, which in turn will shut down the excitatory neuron, which in turn will release the inhibitory neuron, which in turn makes the excitatory neuron active. So this loop, the most natural thing it will do is to start oscillating back and forth because one excites the other inhibits the other. Everybody cool with this? So this is there. Seems like a bad thing. You want to represent the other thing, but instead you're generating oscillations which have nothing to do with what's going on outside. Why would the brain do such a thing? And why would the brain generate spikes? And why waste energy with all this and do something that's totally unrelated to representing the stimulus, which is generate some random oscillation which has nothing to do with what's going on. So let's again do this thing, and we become physicists once again, that, aha, if I can do this, I have a neat segregation of jobs. The excitatory neurons, they're keeping track of whatever is the stimulus space. We're just going to call it space because place cell is space, but it could be whatever stimulus you want to represent, images, this and that. And it's known that whenever you present images, for example, in visual cortex, it's the excitatory neurons that care a lot for exactly image A versus image B. Inhibitory neurons don't care as much. Inhibitory neurons, at least in hippocampus, generate this very strong rhythm, and rhythm is just a clock, so rhythm keeps track of time. So now we have this neat segregation of jobs. One type of neurons keep track of space, other type of neurons keep track of time. Interesting. So let's see if that can solve the problem. And we are just going to go through it together. And for making it simple, I'm going to discuss a model where I'm going to get rid of this connection for the time being. So I'm starting with asymmetric input rather than symmetric, just to make things simpler. And even though this recurrent input exists, I'm going to even get rid of it. So now this neuron gets input about space and gets clock-like input from inhibition. Just two standard things that every neuron gets. Everybody cool with this? So are these oscillations available? Yes, they are available. You put an electrode in the rat's brain. This is time in seconds. And this is the rat's running speed to show that he's running. And now what you see is that this voltage keeps fluctuating pretty wildly, and it's more or less about 10 times a second. Quite noisy, it's not periodic, but it fluctuates. So all group of neurons are synchronously active at more or less 10 hertz. So that oscillation that we wanted is available biologically all the time. And in fact, people have found that if you do some pharmacological manipulation and get rid of this oscillation, the animal cannot learn. Even though neurons are firing, even though this Hebbian plasticity is intact, you simply get rid of the oscillation, animals cannot learn. And in fact, one of the standard tests or biomarkers or treatment for Alzheimer's disease is to give some drugs that can boost these oscillations. This is also seen in human beings. So put some electrodes on the skull, ask them to do some memory task. Whenever the oscillations seem to be stronger, that's when the performance is better, especially in this part of the brain. So there's some solid link that there's something to do with this. Yes, please.
I'm a little confused. So
Yeah.
this rhythm, does it correct for when the red-blue marks are out of order or out of pattern?
There are lots of situations in which they happen. There are situations in which there's some error that happens or mismatch and so on, but it's a wide variety of things.
And these rhythms help correct the error, or?
We haven't stated that. All I'm saying is that even when you do something totally vanilla, so I'm just walking here. I just walk from here to there. There's nothing special. If I had electrode in my hippocampus, I'll see a rhythm. And the rhythm tends to be fairly periodic, not perfectly periodic. It's kind of there, but not perfect. But you're going to simplify that, and then we're going to look at all the noise and all that stuff. And it has all kinds of frequencies. It's not just one frequency, right? But we're just, again, going to start with, just want to show that this does happen biologically, and now we can go with it, okay? So whatever ingredients we need, which are there, we are going to put that in. Everybody cool? So now let's go with the simple model. So here is my neuron, rat that's running around. There's just one cell. The rat's running around from here to there. Y-axis now has some current that is getting injected in the neuron, doesn't matter, some picoamps, nanoamps, depending on where you are. And this neuron is getting this excitation right there that is asymmetric. So we are assuming that network is already learned. Bear with me how the learning happens in the next step. So that's a smooth function. And then on top of it, the neuron is getting this inhibition as well, which is also a smooth function. Now, you can say, wait a minute. Why is excitation a smooth function? Because neuron is getting input from a lot of neurons. Each spike is punctate, but the sum of all that activity is what activates the neuron. From one neuron's point of view, all the inputs sum up, it's smooth function. Similarly, neuron gets inhibition from not one inhibitory neuron, but a lot of inhibitory neurons. All of them we can sum up and say that too is a smooth function. smoother than one individual spike. Some bumpiness, but we are going to ignore it. So far, everything cool? Now I'm going to pull a hat trick, like I did last time to show causal learning. So if any of these assumptions, or not rather assumption statements are unclear, the next step will sound like magic. Yes, please.
Yeah, if there's a lot of inhibitory neurons hitting this thing with these kind of oscillations, wouldn't they have to be exactly in phase? Or
Yes, they are actually.
they are.
They are in phase, and there are solid
Otherwise it
mechanisms. Exactly, otherwise it won't work. Very good point. So all the inhibitory neurons that are giving that inhibitory input, they better be in sync. That's exactly right. And all the excitatory neurons that are coming in, they better be sort of out of sync a little bit to create two different smooth functions. That's right. And that happens. I skipped over some of it, but if you want, I can show you evidence, but it's there. Yeah.
Why aren't those excitatory neurons that are exciting this neuron inhibited by the same inhibitory?
They are as well, but we're going to ignore that.
Okay.
Gets too complicated. Go with me for now to say there's just a bunch of excitatory neurons doing a bunch of things. Stay with me there, and then we are going to bring that in. You can easily imagine one brain region is paying attention to this, and the other brain region giving input doesn't care for this, for example. So it doesn't get hit by those things, okay? Okay, so now we have simplified the problem tremendously. We now have just one neuron. It's getting just two inputs. One is a smooth excitatory input. Other is a periodic inhibitory input, and both are more or less smooth because they are averages of lots of neurons, which is true biologically. What will the neuron do? Well, inhibition is subtractive. That means the neuron can fire a spike only when excitation exceeds inhibition. Because that's when there are more positive ions than negative ions. It has chance to reach threshold. It's not guaranteed that it will fire its threshold, but it has chance to reach. So at this point, the neuron has no chance to fire a spike because inhibition is that big, excitation is that little. And right here is the golden yellow sunrise. That's the first time in this whole period is 100 milliseconds, it has a chance to fire a spike. And that window remains open for just this period of time, and then the window is shut down again, finished. It has to wait another roughly 100 milliseconds, and once again it opens up, goes for a little bit, then again shuts down, window opens up, and now what you see that because of this smooth function, I have generated a different language. If I want to know where the rat is in this space, I have to look at the very first spike the neuron fired after a period of silence, which is how our words are, right? How do you put my words together? I have some sound and there is silence, and a sound and a little silence. So if whoever is listening to neuron says there was a long period of silence, so the first spike that happened tells me where the rat is. If the first, now let's put this thing to phase. So for example, this phase is 0 degrees, that's 180 degrees, and that's 360, and so on. If the very first spike occurred more or less at 359 degrees, the rat must be at the beginning, just entering the place field. So instead of the rat being anywhere here, with just one spike, I don't have to count the firing rate. If I want to count the firing rate, I have to average over a certain window. That takes time, and we have to behave fast. So with just one spike, I know roughly where the rat is. Then if the first spike occurred instead of 359 degrees, at maybe 350 degrees, then the rat must be slightly further in, and so on and so forth. Right? Everybody cool with it? So by using simple rate-coded neurons, I have generated a code that uses spikes, and it's extremely fast. One spike, and I know rather than average a bunch of spikes, which creates problems. Yes, please. Adding. Just subtracting or adding, that's right. They don't multiply. There is one kind of inhibition that does some kind of multiplicative or divisive, but most inhibition is like that. Because they are negative ions versus positive ions. Okay? So that will do it. So as a result, the very first spike that occurs here is here with respect to the peak. Keep track of it. This spike occurred here, and then occurred earlier, and then it starts to appear all over. So this whole phase space is used to encode where the rat is, based on spikes now, using smooth inputs. Because every neuron needs dozens of inputs to do anything. Now, how does it solve the problem? Well, the first thing is that the two languages the neuron uses to communicate, one language is the average activity, the smooth function, that's a smooth input. Smooth input comes in, it generates two languages. One is the average activity, because here, because the excitation is so large, there'll be more activity as well. But for that, you have to wait. It's good, but you have to wait to see if they got more spike or not. So there'll be more spike here net. Here, the net spike will necessarily be less. So now what I have generated this is by subtracting away excitation, right? There's a whole bunch of excitation. I got rid of it by inhibition, seemingly waste of energy, because all this excitation that came in here, I'm throwing away. All the excitatory input that's coming in here, I'm throwing away, seemingly wasteful, because all those neurons wasted their effort sending excitation, which costs energy. So by throwing away this input, but in a selective way, I improved my representation by getting two languages that tell me where the rat is. One language says, give me the exact phase with respect to this. Other says how many spikes were there. Just have more spikes, then you're at the end, less spikes are at the beginning. So something useful, but it solves the problem that we started with as well. So now let's imagine there was a second neuron who was slightly shifted like this, but we are making it asymmetric. We're going to make it symmetric in a second. Now the cool part is that because of this particle in a periodic potential, that's what more or less this is, but the excitation is not flat, it's smoothly changing. Because this neuron is after this and it's also asymmetric, this neuron will always come out of inhibition after that. So in every cycle, the blue neuron has a chance to fire first. Whether it fires or not, we cannot guarantee. But if the blue neuron fires, the green neuron cannot fire before that. Can do some perverse things such as wait too long, but it has minimized the chance. So now what you're going to get is that even though your behavioral time scale or which your cognitive time scale or neural representation time scale are slow, of the order of a second, by using inhibition, which is smooth, and excitation average, which is smooth, you're creating spikes which make sure that the two neurons are reflecting the same behavioral time scale, and that, as it happens for the oscillation, this being 100 milliseconds, this is about 5 to 10 milliseconds, which is what the learning rule requires. So you're going to get the same thing repeated many times, and now the problem that I told you, that you want to make sure that whenever the blue neuron fires a spike, the pink neuron should fire immediately after that. That's much more likely to happen. So now when you want to pay attention to something, all you do is make those neurons active, and you'll start learning those sequence of words that are coming out, and you'll tune out other things, inputs which don't have that. As simple as that. And that explains why when people have to learn a sequence of words or something else, if there is oscillation, their so-called working memory performance goes up. It doesn't prove it, but it makes it plausible. Yes?
But the neuron doesn't have to fire at the intersection of the graphs.
That's the first time it can fire.
Between the
Exactly. The neuron can fire only at this point, not before that. This neuron here, the blue neuron, cannot fire in this time period. That's right. It has this interval, it's fired, that's right. If you want, I can go through why. This part you can say, all right. If you haven't thought about this, don't listen to me right now, but this is just for you. You can say, well, at this point, the whole sequence is reversed. It's a bad thing. That because the picture that I'm drawing so far ignores the recurrent inhibition. This was a simple picture I drew, and the solving equations not. I'm drawing what happens when this is not present. This was the picture. But now, as soon as this neuron fires a spike, it'll activate inhibition, it'll shut it down once again. Take some time. So the later part will not happen. So what happens is that as soon as the neuron generates a spike here, at this point it can generate, it excites inhibition, and now inhibition cuts off the probability of firing any further. So this part will not happen, roughly. And actually, there are other mechanisms that biology has implemented too that make sure that that's what will happen. Yeah?
I guess I think kind of putting a spectral analysis view on this, it seems like the rate at which the red line is moving, right, could be optimized given the difference.
Exactly, that's exactly right. So what you can now do is that there's an optimal, now we can play with this to say, okay, this will at least solve the problem, but can I make it optimal? That means that depending on the window of learning, which changes with development, infants have very wide window of learning, that 10 millisecond window we talked for infants is very wide. Then, depending on the bumpiness in this picture, which I ignored, depending on the length of this excitation, I can come up with an optimal tuning frequency. And I'll show you evidence that that actually happens incredibly. Yeah.
So maybe I misunderstood, but the way in which you described inhibition, obviously in this cartoon, is periodic.
Yes.
But then you just described it as responsive to the excitation signal. So,
Yeah.
I mean, it's not periodic?
That's right.
So which is the
Very good. This was, this was simplification. As I showed you in experimental data, the inhibition is not very periodic. The total activity is not extremely periodic. It's doing weird things. Now, if you go back to, you're absolutely right. It's not very periodic. But now when we look at this, now let's take a deep breath and say, let's make this signal aperiodic. Instead of every cycle being the same length, every cycle is of different length. No problem.
Right, although in that other figure, it also wasn't obvious from that figure that there was a counter increase in inhibition in response to
That's right. That was the average activity.
Okay.
That was just the average activity. I'm simply showing you, without going through dirty details and so on, that in this picture, imagine this period is this much, now that period is much wider. Makes no difference. The same idea will work. As long as it doesn't become very, very narrow. So if this whole thing became like that wide, then it doesn't work. Just bounded from below, how narrow it gets, but it can go as large as it wants, it'll still work.
So what would that be a problem? Because I mean, if
Because
it
then
just
the timing
spiked,
will not
if it
work.
instantaneously spike, then
Spike will work, but we know the learning rule constraint. The input and output neurons have to fire in the same order within 10 milliseconds or so. Otherwise, things cancel.
okay, and cell 2 is the
Cell 2 is the second neuron that's coming after this. So cell 2 has to come. So a single neuron, there is not a problem. There are additional problems.
so coming after it. It's one that cell 1 is activating. Or
That's right.
Okay.
What we are trying to do in this simple version is to say, okay, on time scale of behavior, first there was blue neuron, then there was pink neuron. And in that simple two-bump model that I showed you, you'll grow a synapse and sequence will be learned. What I'm showing is that if neuron is actually spiking instead of smooth neuron, this will still work. But on top of it, you got a control of what you will learn and what you will not learn. What will you ignore and what you will learn. Yes?
Will the tuning frequency change after this?
We're not going into that. Tuning frequency does depend on things, but that's another talk altogether. One sentence summary is that the faster you run, the faster the frequency, so you're computing things faster. So that happens. How that happens, long story. Yeah.
It looks to me that what you described here in engineering terms, it's A to D converter.
Yes, that's right. But A to D converter with variable threshold.
Yeah, but there
My
are some.
threshold is changing.
Yes, there are some.
That's right.
There are different ones.
Yeah, that's right. It'll be awesome to think about that. This can be implemented in hardware. So this can be pretty useful. So let's talk about that. Yeah.
This is a crazy question. So the reason our cell phones don't trample on one another is that they keep silent at random intervals within a second. So this randomness of this input
Yeah.
modulating red curve,
Yeah.
could it be that way you're enabling several simultaneous
It's a great question. I have thought about this, and there is some evidence that that's happening, which is, let's talk about that. Very good question. Yes, actually, so there is some thought about that, but let me not go into that. I have only thought about it partially, so let's talk offline. Okay? It will take us quite far into different oscillations. So now, we started with a question to say if neurons have to learn And if they are smooth functions, they can learn, but if they're spiking, everything will break down. We now came up with a mechanism to generate spikes and include inhibition, which artificial networks don't have. They don't have oscillations, but by putting this, I generated something like attention, and something like this will be learned and that will not be learned. Very simple. So first of all, does it work or not? Because I am still a physicist. Yes, please.
So, okay, the blue curve and the pink curve, those are not learned, those are just excitations.
That's right, that's right.
And so what's being learned is to have these spikes happen in a certain order. But where does that learning,
That's right.
where does it,
That's right.
where
That's right. So
is it stored?
that's exactly right. So what is being learned is not this. As long as this was the average excitation for one neuron and this was the average excitation for cell number 2, this will just happen. The learning will happen in terms of the synapse from this neuron to that. Because remember, this was the blue neuron that was active first. So let's go back to that picture. Remember, we started with this. So there is this neuron and that neuron. And if without this oscillation, the timing will be weird and that learning will not happen. That this came before this, in this situation, it will not work. Because you could easily have a situation like this. I solved that problem.
But you're saying that because of this order of firing, a synapse is strengthened. Is that what you're saying?
That's right. Because the learning rule that I described last time is such that if instead of this, the previous thing happened, which is what I have now generated, that if red neuron fired before this pink neuron, then that strength will be made stronger. And then this strength will be stronger, and this will be stronger, and so on. So what I'm engineering is, we know the biological learning rule, which is what I went through last time through NMDA receptors. They say that on a scale of 10 milliseconds, if neuron A fired before neuron B, on a scale of 10 milliseconds only, then A to B will be strengthened, but B to A will be weakened. That's the hardware learning rule. We didn't know how to come up with how to engineer that biologically, because that's no way to guarantee. You can easily have something like this where that's not allowed. So I showed that that will work in a network, and now I came up with a mechanism to generate this. To say that that learning rule will now make sure that in these two neurons, from this neuron to that neuron, a growth process will happen, and that will generate anticipation. Does it happen? I need to show you, but that's a plausible mechanism so far. Okay, so learning happens in the synapse. This is just network will do. Even on a random piece of paper network, that's what will happen. Now if you convolve that spike timing by learning rule, then synapse from here to here will get stronger, because the green comes always after yellow, but the reverse synapse will get weaker, because that's a hardware learning rule.
And what does that do for you? I mean,
that's right. It induces an arrow of time. It says that the arrow of behavior, which is very slow, when you go from here to here, this window, by the way, in biology is 100 milliseconds, as I wrote. So this whole thing is 1 second. So every neuron is representing information on a scale of 1 second. But the learning rule is precise only within 10 milliseconds. So by doing this, what you are doing is that the behavioral level sequence is being replayed on the time scale of the learning rule. That's number one it does. Because otherwise it won't happen. The two Gaussians that I showed you, that was not being replayed, right? So let's go through that again. So if I look here, in this picture, this is one neuron spike, that's other neuron spike, but there is no guarantee that whatever happened on the behavioral scale, which is red before blue, that was not happening on this short time scale of learning. Learning only cares for what happened in 10 milliseconds. Here, the red is blue is sandwiched between the two. Here, the blue is too far. And here, blue comes before red, because this will happen just by chance.
that part I feel like I understand.
Yeah.
But, so now I'm a rat walking along here.
Yeah.
What did I get out of this learning process?
What you got out of this is that your neurons, if they were learning, as we know by this biological learning rule, the neurons are able to now reliably learn the same sequence to say, first time I went from here to here, so make this thing more asymmetric. That will make, in turn, make the replay better, which in turn, so it will be a feedback loop, right? So what you're doing is you're learning this to that. So if you were now to start with a symmetric connect inputs like this, half of it will do this, half of that will do slightly poorly. And as you start repeating this, you will learn that if I went from here to here for whatever purpose, maybe I want there to get a reward, maybe I just lifted my hand, maybe I was running away from tiger, there are a variety of reasons. But that behavioral time scale to say I went from here to here reliably, that information got stored in the brain. To say when you're here, that's the most likely direction you go. That's what got learned. Because that's what this will do. This synapse will get stronger. As a result, this will start to become steeper, and as a result, this process will accelerate. Clear or not? Unclear.
Officer, so in each place, I have a neuron that's doing
Several neurons.
several neurons.
Like 2 neurons. At this place, there are 2 neurons that are active.
But what it seems to me you're saying to me is that I have these neurons that fire at places, but I don't have a map as to what's.
Exactly. I haven't stored that information.
So this is allowing me to start storing.
Sequences.
This is near this, but also that I tend to go.
Exactly. That's exactly right.
So what if I, tend to pace around and go back and forth?
I'm going to show the result.
Okay.
So let's think about that first and then show you the result. So what will happen now? So let's look at this.
Can we go back to the previous slide of the 2 slopes, just 1 second?
Yeah. Yeah.
So I'm wondering.
If you don't mind, let me just finish that answer
Yeah, go ahead.
and then I
Yeah,
come
sorry,
back to
yeah,
this.
yeah.
So now what's going on is that as we know that learning rule says neuron A and neuron B, if A fired before B within 10 milliseconds, make the synapse stronger. We agree? So that process will be repeated now 8 or 9 times. So as soon as you go from here to here, the connection from blue neuron to the pink neuron will become strong because it's been reinforced 10 times. But the connection from green neuron to the blue neuron will actually become weaker. So you have now learned that when you are here, the most likely thing to do is go that way, not that way. In a one-dimensional world, you go either this way or that way, no other choice. When I walk repeatedly, this will become stronger, and now this pink neuron will start firing earlier. That's your anticipation. So when you are here, the pink neuron was never active, but now because this strengthening from here to here, this pink neuron will start getting active, and that is your tiny bit of imagination of where the hand is going to be. And then the next neuron takes over and the next, and now it's a whole relay process. That's what it does. And that's what I showed neurons actually do. In the previous slide that I had last lecture, neurons actually within several trials, they start to represent the future location rather than the current location. Clear? Not yet?
No, that part is clear, but then I don't only go in one direction, right?
Because that's right. So in this simple model, we invoke the property of the neurons that these neurons are active only in one direction. They're not active in other direction. So let's imagine I'm walking in a circle. On that circle, I'm going in this direction or that, and different sets of neurons are active. How that happens, how we break the symmetry, longer story. Yes, please.
So 2 questions that are separate from this domain. So one is, let's suppose I'm trying to just figure out whether inflation is higher in one region than the other. There's
Yeah.
a blue curve and a red curve, but that data is incredibly noisy,
That's exactly right.
right? It seems like I could use this as some sort of
Exactly.
criteria to determine, okay.
That's exactly right. And you can now put different, so you don't want to do supervised learning, you want to do unsupervised. You say, okay, this data set I trust more. On that data set, I put a stronger amount of this oscillatory stuff, which throws out some and keeps some. Other data set is less reliable. I put weaker amount of it. Because there, all those things will start canceling, and you want it to cancel because you know the data is unreliable, which is like paying attention to different things that you will learn.
Okay, and that's my second question. Because when I think in the social realm, I can think of, you're calling this inhibitory curve, I can think
Yes.
of my attention just kind of going up and down. So if my
Not attention is going up and down. What is known is that as you start paying attention, neural activity itself starts to oscillate in the brain. We know this from lots of literature, a giant amount of stuff. Okay? So that's what you did. So this network can do that. So now, I want to show you one or two things because I'm running out of time. So I would like to show you some evidence that this does happen. If you think about it, if you try this in some networks of your favorite choice, you want to know whether this is effective or not. Did it actually work? Because in real data or networks, all kinds of other things are also going on. So one major prediction out of this, apart from learning these sequences reliably with spiking neurons and so-called attention and modulation of selective neurons, is that neurons should be actually representing information in two different languages simultaneously. One language is how many spikes. There are a lot of spikes towards the end, very few spikes at the beginning, and the other language is the phase at which the neuron fires a spike. And those two languages are not independent. They're inversely related. It's the same process in the end. Wherever the net excitation is high, the firing rate is high, but it comes out of inhibition sooner. Wherever the next excitation is very weak, the firing rate is low, but it takes a long time to get out of excitation. So these two languages, which people have debated for the longest time, that how does the brain store information? What's the language by which neurons communicate information to each other? It says actually, in this scenario, neurons will talk to each other using two different languages. One is the total activity, and the other is the timing at which it fired spikes, and the two will be anti-correlated. So both of them in one shot. Does it happen? So I'll just show you one simple example where it happens. So this is actual data now, not model. X-axis is the firing position of the rat. Don't look at the top yet. Y-axis is the firing rate of a neuron. As we showed last time, they tend to have this ramping shape with some bump and noise and so on, of course. Used to be there, that's still there. But then on second plot here is the other language, which is the phase of this oscillation at which the neuron fired a spike. We agreed that the lowest inhibition level is 360 degrees, highest inhibition level is here, and what you see is that whenever the net firing rate is low, the phase is all the way to 360, and when the firing rate is high, the phase is very low. So the two are indeed highly correlated like that. So, and then you can do quantification, you can test many, many things, and it seems to work out. Everybody cool with this? Did the test? So I'm not going to go through different tests, and I want to show you one more thing. This is all, 20 years old. We have not been sitting idle since then. Has keep working, so there's more and more evidence that's coming through that this works in many systems. So you can have an unsupervised way of paying attention and an unsupervised way of learning sequences of predictive coding or successor representation purely biologically. So let me go slightly further and end with this thing. So all this is biological, and now we want to go further. First of all, any questions remaining about the previous part before I end with this thing? So now, everything that we did, we went from smooth point neurons to spiking point neurons, but then there's this other little fact, which is older than both of these facts, that everything we're talking about is about this, the cell body. This is one kind of neuron. This is gigantic. This, as you can see from scale bar, 1,000 micrometer. And for more than 100 years, we identified different neurons based on these dendrites. So all this picture, it's not that the picture is kind of grainy. All this varicosity that you see here, those are synapses. That creates a little bulge. So this is like 10,000 synapses. This is neuron in the neocortex, layer 2 or 3. This is a different neuron in layer 5. Notice these dendrites have a very different shape. This looks like, I don't know, one kind of tree, and that looks like an upside-down palm tree. The base is nothing, and all the leaves are here. These are all dendrites. These are neurons in other brain regions, CA3, which looks like a mesquite or some plant. And this looks like something else. So people actually noticed this 100 years ago, that if you go and look at any brain region, the neurons have the dendrites have very typical shape. very reliable, and the shape is preserved across species. So we go to cerebellum, the neurons all look like Egyptians. They're all like very planar. These are 3D. And in cerebellum, neurons are very planar, these Purkinje cells in all species, and the shape is preserved, even though the size changes. Layer 3 neurons have this shape, kind of whatever our eyes tell you. The size changes between mice and humans. They become bigger in humans, but the shape remains. There's something about the shape of the dendrites, where all the excitation is, that is preserved. So why is that? Why did evolution create unique shaped dendrites, which are preserved across species, and they are so big? Because real estate is big, you want to minimize the amount of hardware you're carrying because it consumes energy to maintain. Why make it so large? Why not make a little spherical neuron, have all the input come in? In fact, the problem gets worse than that. If you have one excitatory input that comes in here, by the time it travels all the way down here, it can easily take it 5 milliseconds, and you know the learning rule, it's very precise. In fact, it gets even worse than that. What people have found is that once this neuron generates a spike, that spike starts to go back in the dendrite, it has to go to that synapse and remove its magnesium block in order to learn. But unfortunately, when the spike goes, it's going to remove magnesium block of everybody in between too, because just voltage traveling. Those other synapses need not have activated. How is it going to keep track of it? The problem gets even worse than that, that when you start going here, biologically, evolution has made sure that even though the learning rule says spike has to come at the right time after the input, evolution has also ensured that the spike, when it goes down the axon, it travels noiselessly. The whole voltage goes down the axon very, very fast. But when the spike goes back in the dendrite, its amplitude actually starts reducing, dissipates. And by the time you go halfway through, it's ineffective in removing that magnesium, and there will be zero learning. So once again, that very nice story that I told you started with smooth neuron. It worked, but then we realized spikes create problem. Now we realize spikes can work, but give you some advantage, such as attention and selective learning. Now we need to think of this. Why this? Why neurons have gigantic dendrites? Why is the synapse that learns is so far away from the soma? How will this cell body know which synapse activated it to do the right credit assignment? There are lots of problems. So this could not be solved for the longest time. Everything that we measure, because we can put electrode only here, and we can only listen to spike here, this you can't measure. So nobody practically knew what happens. So this is where we decided, all right, let's play around with it. So what happens is that when you put an electrode, it has to go very, very close to this, and the brain is sloshing around. When I just did this, my brain sloshed around by easily 50 micrometer. And these neurons are jam-packed. There's no empty space, unlike this picture. Is another neuron here. If I put electrode here, it better be flexible. If it doesn't move with the brain, you're like sticking a knife and moving the jello. Everything around it is dead. So you got to develop some new technology. So you got to make flexible electrode, number one, that when the brain sloshes around, when you move, when your heart beats, the electrode moves with it. And then we created a little gap between this electrode. We call them chopstick electrode. So the dendrite can get trapped between these. So we tried to make electrode like that. So some amount of it worked, and a lot of it didn't work. So I'm going to tell you the story when this is very primitive technology. We are trying this. I haven't figured out whether this ultraviolet light-based blasting of the gap, did it really work or not, or whether something else worked, but 2 out of 100 times this technology worked, and that's how new technology works. Failure rate is 98%. But 2 out of 100 times this worked, and the signal was quite interesting. So we figured out how this works. And when I say we, this fantastic student will soon be in the job market. So those who are listening, pay attention, hire him. And now I have to make sure the video is turned up, audio is turned up, because you have to listen to this. So when I started this lecture, I played you the sound of single neuron spike. Hopefully you remember that spike is like almost 1 kilohertz sound. This is when the signal doesn't work. You don't hear anything. You'll hear a little shh kind of sound. The audio is not loud enough. Let's see if this works. I'm going to show the real signal. So now comes the real signal. This is 2 out of 100, enormous amount of effort. I'm going to shut up rather, and let's listen. Do you see that signal? Does it sound like the signal I showed you before? What's the difference? Much more bass, much more low frequency. The bass here is not that powerful. This is the signal recorded in the dendrite. This is the standard extracellular recording the tiny little spikes. But when this grabs onto the dendrite, you get these gigantic spikes. So the first thing is, like many people had suspected, that what if the spikes are not generated only in the cell body? Maybe spikes can be generated in the dendrites too. And what we found is that's actually true. Dendrites generate a ton of spikes, and they are different rather than the ones we see. Let me skip these things. Let me skip all this.
Sorry, the bottom curve is the soma and the
Not the soma extracellular, that's exactly right. So bottom curve is the signal measured from the cell body, but with the standard electrode outside, so the signal is much weaker, otherwise soma signal is even bigger. This thing is from the dendrite, but it's grabbing on, so signal is much bigger. And a couple of other things happen as well, that's why the sign flipped and all that, which I'll skip for now. It's in the paper. And the signal is,
Quick question.
yeah.
What is so, obviously there was some interesting
Yeah.
shifts. I mean, not in this
Yeah.
prior one. So are those associated with behavioral or
We're coming to that.
perceptual events?
Coming to that, yeah, we're going to come into all that. So I first need to demonstrate that there is a new signal in the brain which we haven't paid attention to or we never measured before. It's a totally different kind of signal, and it points to a different kind of mechanism. So this is one dendritic spike. It generated, we have reasons to believe it started here, and I'm super proud. Imagine this is single trace in a freely behaving animal. Amazingly clean trace. You can detect exactly where it began. This is the average and 95 confidence interval of this recording for 26 hours. This, you're supposed to fall off your chair if you know how to do brain recording. If you patch a neuron and measure the voltage of a cell, you can do it for 20 minutes and the cell is dead. This thing works for 4 days. This is large amount of signal. This neuron, we got some 64,000 spikes in this 26-hour period, and the activity, it was very reliable, very broad spike. It was not 1 millisecond. It started off here, but lasted up to here about 10 milliseconds. So the spike is very wide. You have to keep track of that learning window, which is 10 milliseconds. These spikes are very wide, 10 millisecond wide, unlike the somatic spikes. And they dominate this. The number of these dendritic spikes are 10 times as many as the number of spikes in the soma. So I'm going to skip all this as well, because there are many shapes, and those shapes will influence. There are different layers. These are different shaped spikes. And in all of them, if you measure the firing rate, these red are these dendritic spikes, blue are the somatic spikes. The dendrites are generating 10 times as many spikes as the cell body. Massive amount of spiking. So everything we have been thinking about to say here's a somatic spike, it does something and so on, misses basically 99% of what's really going on. Let me skip that as well. And I want to come through the final point. So then what you notice is this. Let me see. So here is the voltage of one dendrite. Here's the voltage of another dendrite when the animal is doing nothing. Because when the animal is running around, lots of things can be going on. What you notice is when the animal is doing nothing, there are gigantic fluctuations in the subthreshold voltage, and on top of it is a spike. Now what you notice is the coding mechanism in the dendrite is completely different. Because what I showed in the very first lecture, the standard textbook idea, which is still true, that for the cell body, the subthreshold fluctuations are about 10 millivolt, the spike is 100 millivolt. That's why we called it all or none. That's not true here. The subthreshold fluctuations are always bigger than the spike amplitude, all of them. So x-axis is the subthreshold dendritic membrane potential voltage range for a different source. This is the dendritic action potential amplitude. This is the unity line on a log-log scale. Every time the subthreshold voltage is bigger in the dendrite, and it actually modulates the spiking probability. So if you plot here the subthreshold voltage magnitude versus the firing rate, it's modulated in a smooth sigmoid fashion rather than a strong sigmoid fashion. So there is the fundamental code that's going on in the brain in majority of the tissue. In the cell body, it's digital, zeros and ones, but 99% of the tissue in the neuron where the signals are coming in, which are the dendrites, there the signal is hybrid. The analog component is very large, and that analog component does generate spikes, and the spiking probability is modulated by the analog component. So the fundamental coding mechanism in the brain is actually hybrid. It's analog and digital at the same time. And that will influence a lot of things, and let me just finish off by showing you one thing. Any questions before I go on? Yes, please. There's a hand there too.
Say, do you want to push anything here?
No, this person should just unmute themselves and speak.
Okay.
You can ask your question.
Can I ask? Yeah, is it possible to obtain this phenomenology using simulation with Hodgkin-Huxley model?
Yeah, we did that as well. It's in this paper. If you use Hodgkin-Huxley in the right, the certain distribution of voltage-gated sodium and potassium channels, you will get this, and that's some of the part that I skipped, which is right there. In a biophysical model, you will get this. So look it up. It'll take me too long to say how it works because it's too complicated. The short answer is yes, it works. Did I answer the question? Anybody else? Seems like, yeah. Oops, sorry. It's cool. It's cool technology. Yeah. Go on, please. Whoever is on Zoom. Nope, you're gone. All right.
I'm going to ask my question quickly. So, something comes in over synapse, and an electrical signal travels down from the dendrite to the nerve cell. So, what are you measuring the dendritic voltage?
Let's good question. Thank you for asking, because one gets used to what one is doing, and then so let me go back to this picture. So the standard idea was that here are all these little bumps are synapses. That signal comes in, goes all the way to the cell body, then the cell body decides whether to generate a spike or not, and that carries all the signal. What we find is that's not really happening. What happens is that imagine, so we are trapping one of these branches, or so we think. We can't prove it, but that's what we think we are doing. So when you trap this branch, what's going on is that this little branch here itself has synapses. And these branches, it turns out, are much narrower. As you can see from this picture, this is pretty big, this is less wide, and this is even wider, just like the branches of a tree. As you go farther and farther, they get narrower. So what happens is that when the synaptic inputs arrive here, the resistance of the dendrite is higher, because the narrower the dendrite, higher the resistance. And we know that these dendrites also have the right hardware to generate these spikes, but they are different type. They are slower, wider spikes, and so on. So a small amount of synaptic current that comes in here will add up and generate a spike of its own, because the whole thing is very narrow, so it doesn't need as many spikes the soma to raise the threshold from -65 to -55 and generate a spike. That's exactly right. Within that thing, you are generating giant depolarization and giant amount of spiking, 10 times more in many of these branches. How many we can't measure yet, but as many as we have measured. And then once it comes here, now all of a sudden it's like a tiny river, that's a stream that's going in and makes a big fat river, it gets dissipated, because this is much wider. So it doesn't really go there. Once it comes here, the whole thing is massively dissipated because the impedance is reduced massively.
So
It remains local.
it's telling things to the other synapse.
Exactly. So what happens is that here, imagine there were two synapses here. The same cartoon that I've been running with, instead of one synapse, if there were two synapses, one synapse here and other synapse here, they will not be able to communicate with each other. Not only through dendrites, but also backpropagating action potential. But if there were two synapses right next to each other, then or 5, let's say, if two synapses got activated, that will rapidly depolarize this, kick out the magnesium block of the remaining one, recruit them, and now you have mapped memory sequences that happen in behavior right onto tiny branches. So what this says is that the fundamental unit of memory is the segment of dendrite. Each segment of dendrite stores a different unit of memory, a different sequence of memory rather than just one single snapshot. That's what this does. So it says that the reason these neurons have these interesting looking dendrites and so on, because these dendritic segments store one simple unit of memory. I may have even written this down somewhere. Yes, please. That's right. That's right. So I don't know if I have it somewhere. There you go. So one can say this in a very simple way to say that if you start with neurons like this, the standard idea is that the input comes in there, then it has to travel to the cell body, and then it has to generate backpropagating action potential. That's not going to work because of a variety of reasons that I mentioned, it'll dissipate and so on. But if on the other hand, you have two different synapses, one synapse here and a different synapse there, they are not going to add up because they are too far. But if those two synapses are on the same dendrite right near each other, then the voltage will summit, it'll cross the threshold for spiking, and now they'll remove each other's voltage block. Now you have stored the memory right into the little segment. So the way I stated this in my ultimate chutzpah, the standard learning rule is that we started with is that if a neuron A repeatedly fires before neuron B, the synapse from A to B is strengthened. That's the way the rule that I've been working with. But what the dendrites do is something else. The new learning rule says that if synapse from neuron A and B onto a different neuron C will be strengthened if A and B are repeatedly coactivated and if the synapses are located near each other on a dendritic branch. That's an additional constraint. It provides additional advantages, and I can show you some advantages, but I think I'm running out of time, so I don't want to go too long. I see somebody waiting at the back to make sure that I don't go too long. So I'll rather take questions, and if you want, I will show you. I didn't answer a question about how these dendrites represent signal. Maybe I'll show you very quickly. It all comes down to predictive coding. So let me just very quickly show you that if I can find it there. So what then we did is to look at the signal. So it turns out that these neurons are coding what the animal is going to do next, literally. Their cell bodies. So in the future, whether the rat is going to turn right or turn left or go back and go forward at a high speed or go forward at a low speed. So these neurons, we know this somatic spiking activity encodes the rat's future set of actions, future in a scale of about 100 milliseconds. That's why it's a source of predictive coding. It's right there in the parietal cortex. It generates funky maps and so on. And don't look at all this. I was planning to go through it, but we ran out of time. That's okay. This is the spike timing with respect to behavior. Negative means the spikes are predicting, positive means they are not predicting. And what you see is that the somatic spikes, they are highly predictive. Some of them are coming after, but majority of the somatic spikes come before behavior. So the somatic spikes in a cell are predicting, but these dendritic spikes, they don't have much of a predictive power, nor does the dendritic voltage have predictive power. So a single cell is doing predictive coding. All these dendritic inputs arrive, and the reason the cell has so many dendrites is doing predictive coding or successor representation across these different neurons. So each neuron, each branch encodes whatever is that comes a short segment of sequence, and the whole neuron puts the thing together. That's why the somatic spikes are much more predictive than this. I will take all of this with a grain of salt because we only have managed to record 25 of these despite much effort. We need to get a lot more data to really claim this. So unlike everything I showed so far, this is just one publication. But it seems to be supported by more and more papers that are coming out which support this idea. So it's not entirely the only paper. That's why I bothered to share with you rather than not talk about it. Yeah.
Why are there so many more blue dots than red dots?
That's exactly right. Because our success rate is 2% for the dendritic spike, somatic spike is high.
That's your fault. I'm just
Fault,
kidding.
if you call it, that'll be.
I'm just kidding.
That's exactly right. This is new technology. This is all we could do. And I can, to not be entirely be put down, we are the only people who could record this in freely behaving animal. Even since 2017, it's extremely hard. So I'm more than thrilled if somebody comes in and improves this, so the success rate goes to 5% or 50%, I'll be super thrilled. Yes, please.
So amazing. And speaking of mathematical modeling, the success representation work modeling obviously captures your 2002 paper, but are there currently any modeling efforts? And if not, can we try to figure out how to put the dendrites into the picture?
So we have done some of that work. I have not shown you. We wrote several mathematical model papers as well. So the main thing that is different is that the success represent model relies on a supervised learning rule, temporal difference learning rule or something, which we can argue where it's implemented or not. This is purely biophysics. This doesn't require that. This is unsupervised. This is what the hardware does. Take it or leave it rule. So that's the fundamental difference between the. Absolutely. So see which way. So this is what hardware does. The hardware eventually may come up with temporal difference learning rule. How do you connect them? That's a nice project. We have some ideas, but this is just saying the hardware will do this. That's the way it is. So it's totally unsupervised. Yes, please.
Just a general modeling question, but how do you choose which observations are important for the model to reproduce and not reproduce? So for you, spike timing is important. These observations are important, but probably not the number of proteins in a nearby glial cell.
We are worried about that. We are worried about the nearby glial cells. A lot of it goes in there, but it's right now. It's I'm sorry to say, but it's kind of an art form. You read a ton of literature. You get thoroughly confused about this gazillion biological words. And then from that, you rely on your brain to say, all right, out of these things, like what you were saying earlier, rely on the hardware here. So, all right, let me take my initial guess. So I'm going to just pick 2 out of those 100. I have no way to say how we do it. And then you go and question yourself. So, all right, 2 out of 100, I made this smooth model. And now let me bring in spikes. Now let me bring in inhibition. Let me bring in dendrite. And so far, we have not made major error, but I can't give you a recipe. Yes, sorry, you were here first before. Yeah.
No, I want to say something very silly. So the inhibitory parts of actions, are they also kind of local? Or?
Thank you. I forgot about that. So what happens is what used to be believed, once again, let's just stay with this neuron. Where are the neurons here? So what was believed was, first of all, what is clear even today, most of the excitation is here. Most of the inhibitory, this cell body has a ton of inhibitory synapses. So classic idea was that most of the inhibition is right here. But in the last, God knows, 2 decades, it's become clear that there are actually at least 2 classes of inhibitory neurons. One class of inhibitory neurons target the cell body in this segment. Other type of inhibitory neurons specifically target this. Specifically target the dendrites. Now you may ask, then in this process, what's the role of inhibition? Why would I put inhibition on a dendrite? Because I'm right now to add it up. So in the learning rule and the modeling work that we did, is that if I have a long dendrite, and if I don't have inhibition, once this action potential dendritic spikes add up, it'll remove the magnesium block of everybody, and now I have wasted the whole real estate. But if I put some little bit of inhibitory synapse here, it'll curtail that wildfire from spreading in a dynamic way. It'll allow something to go through and some not. So in that sense, the inhibition has a role. Say it's the bunch of excitatory synapses flanked by inhibition. That's a fundamental hardware unit, which is a unit, little segment of delta T. Because things always flow this way flow is very hard. So that's the delta T. And then I can store many sequences based on these inhibitory flanks. So that increases capacity massively, changes code. Because the computation is analog, massive increase in capacity, computation is local, so noise and so on that seemed like noise is now not an issue, it's a signal. So it has lots of advantages, but we'll see if that's really true. This is as far as we went. Yes.
What is the time scale of dendritic influences on learning as opposed to the cell?
Can't do that. No technology to do that. But we know for a fact, there's a classic work by many people. Somebody, Nelson Spruceton, did that work. He does this. So they said, all right, do you need the cell body or can we just stay with the dendrites? So what they did is, and several other people, Aaron Schumann and the others, what they did is they basically silenced away the cell and simply did the goal of pre and postsynaptic neuron only in the dendrite. Works perfectly fine. You don't need the soma. No chance. You can keep asking in humans. You can't do that. This is really hard, yeah. Sorry? They could do induced plasticity. They could induce LTP on the slice. The slice lives for 1 hour, so you can have roughly for 1 hour. But that's where there is a controlled way to say this can work. The idea is that things can work perfectly locally. People have shown that in a slice where everything is dissociated, you activate it by hand, only those 2 neurons, they can learn without the soma involved.
How many times does it need to be activated? Over how many milliseconds or seconds?
I don't remember. It's more or less the same ballpark as this, but slightly wider window because the dendritic spikes themselves are wider, so the window is wider. But the whole idea that the plasticity depends only on spike timing, that was the toy model that I started with. In reality, this learning rule is far more complicated. And we've written lots of papers about it. It depends on the firing rate. It depends on how far it is and so on. So if you're interested in how wide and what timing and so on, we have a paper in 2012, Kumar and Mehta, which looked at all this. And I may have a slide here somewhere to show you how it depends.
Is there an idea of learning rate for the dendrites?
Yeah, there is a lot of it there.
I am going to combine questions about timescales and inhibition and say that timescales have been reached for thanking our speaker and inhibition.